Afternoon, sir.
Good afternoon.
Good afternoon, sir.
Yes, good afternoon. I can also.
Can we start, sir?
Yes, please.
Yes, you can. You can. Sir, we can start.
Okay, sir. Thank you. Good afternoon, sir. I would like to welcome to Dr. Prashant Singh, sir, our great person for today occasion, respected Dean, sir, respected Registrar, sir, respected Vice Chancellor, sir, and staff member and my dear students. This is the time to stimulate active policy for sustainable development of agriculture system based on farmer families and cooperatives. Programs such as Bringing Green Revolution to India and National Food Security Mission have already provided the much needed boost productivity of major crop in several states. I am sure that thrust to increase agriculture productivity will be continued. Now, what is the plant defense? Plants have involved various strategies to defend themselves against herbivorous pathogens. Although some of these strategies are constitute present at all times, others are introduced only in response to herbivorous feeding or pathogen infection. The induction of direct and indirect plant defense in response to herbivory and other biotic stress is well established in wide array of studies have documented such induced defense. To compensate for this vulnerability, some plants appear to prime specific defense in response to environmental cues that reliably indicate an induce probability of attack before they actually experience an herbivore or pathogen. Now I would like to introduce our today's great person, Dr. Prashant Singh. Dr. Prashant Singh is currently working as an assistant professor, Department of Botany and at Banaras Hindu University, BHU, Varanasi. He has strong background in plant physiology, molecular biology, seed dormancy, and plant molecular pathology. He has been recipient for several prestigious research awards, include Overseas Research Award, UK, USDA, NIFA, USA, NSF, BBSRC, UK, and Taiwan NSC Postdoctoral Fellowship awarded. Dr. Singh is also member of the Principal Scientific Advisory Group to the Office of Prime Minister, Government of India, to advise the Government of India on various science and social policy issues. Recently, Dr. Singh has been selected as Fellow of Linnean Society at London. I would like to call our respected Dean Sir for sharing his thought on this special day. Respected Dean Sir.
Thank you very much, Dr. Vikram. And it's a great pleasure for all of us that Dr. Prashant Singh is with us. He's my very good friend, as well as we work together for a kind of the occasions on different occasions, I can say, at the Rajasthan University, Jaipur. So today's topic is very much, relevant regarding the plant defense priming, and regarding the agriculture science, it is very much valuable on the different aspects. Dr. Prashant Singh, as I want to give some information about the Parul University. We are doing a lot under the dynamic leadership of the various dignitaries of the universities, our Honorable President, sir, Dr. Devanshu Patel, sir, our Honorable Provost, sir, our respected Registrar, sir. So we are doing a lot by catering the society for fulfilling the need in the education. We are providing the education in the different fields, mostly in the field of medical science, in the field of agriculture sciences, in the field of engineering. In the agriculture sciences, we are having this agriculture, College of Agriculture Engineering, College of Dairy Technology. And if you see, we are having almost 100 for doing the practicals in the field by the students. Regarding the today's topic, this is very much valuable if we see how that can be applied with the current science in the modern science. So I think with these words, I would like to welcome Dr. Prashant Singh for today's webinar.
So, good afternoon, everyone, and thank you, the Professor Sharma and Dr. Vikram for a very nice introduction. Before I start, first, I would like to congratulate Dr. Sharma for a dean to the new, the state in the Parul University, because I was associated with, Dr. Sharma when he was in Indore, when he was in Rajasthan, and I'm very lucky. I mean, I think it is a very good gesture even for the university that, they hire him as a dean, because he is one of the guy who can change within a very short period of time. So I think it's a very good opportunity even for the Parul University that they have hired the dynamic, simple, down-to-earth, agriculturalist guy, and he has a lot of idea and thoughts, and he could translate very well in a coming future, if he stayed, the Parul University for a very longer period of time. So before this note, so again, thank you, Vikram, for introduce me. I don't need this introduction, but yes, just one sec. So again, thank you very much. And in terms of the current scenario, in terms of when we talk about the agriculture, we know there is a development of technologies has been comes to forward, but still we are not really on the platform that we talk about the sustainable. So in my talk, when I give a talk, so any technology could be stable in agriculture if it is sustainable. If it is not sustainable, it is not going to work, for a longer period of time, right? So there are the number of technologies has been coming every nowadays, but still they are not sustainable. So we need to think into that perspective. So let me share my slides first and let me know if you are able to see. Okay, just hold for a sec. I need to minimize this. I need to take this back. I need to go here. Present now, window. Yeah. Is it visible? Can you see the slides?
Yes, sir.
Right. So I have given a term that's called the green vaccination. And I really like the term because
Dr. Prashant, it is loading. It is loading.
Okay. Is he okay now?
Dr. Prashant, it is loading.
Okay.
I think it is now. It is okay. Now it is okay.
Is it fine now?
Yes. Okay. Carry on.
So the talk is based on this, the green vaccination. And as I told you before, there are the number of tools for this, the agriculture. But when we talk about agriculture, we need to really fit the idea that it has to be sustainable agriculture. And in the talk, I will try to emphasize and I will try to convince at the end that this technology that I would call that the green vaccination, it could be one of the niche or one of the platform for take this further on for the sustainable agriculture. We know that with the UN, there are the number of SDGs, and even in the government sector, even I had a discussion with the PMO's office that we were working on a number of these SDGs, like the poverty, the good health, gender equality, the climate action, the biodiversity. One of the important aspects of these SDGs is zero hunger. And this is very important in current scenario, that we need to feed billions of people across the world to bring this SDGs. And I was the member of number of these the meetings, even back home, even in overseas, in states, as well as in UK. And we talk a lot of these sustainability, whether it is the soil health, is the forest health, the clean air, ocean health, the food waste. But right now, my talk would be based on zero hunger or the global hunger. We need to find a solution for this, the food security problem. And as I told you before, I was a member of these summits and the meeting where people talk so much about this global food security. But to be very frank, still I'm not satisfied that if we are able to solve the GFS, that is the global food security, we are able to solve the problem. The reason behind that, it is not solving the global food security. It has to be sustainable global food security. Unless and until it is not sustainable, it means the healthy food for all, it is not going to work. So there's a reason in the current scenario, when we talk about GFS, we need to think SGFS, sustainable global food security. And as a, the plant agriculturist, the plant immunologist, we need to really understand what exactly is sustainable. So in my definition, there are the three P's like my name: people, planet, and plant, or the people, planet, and profit. We need to take all these three pillars along with our research work, and if they are three together, then any technology would stay for a longer, there could be durability, and it could be sustainable. I could explain in a very simple layman language. Suppose if you bring a very nice resistant variety, again, wheat pathogen. If this variety has an impact on biodiversity, it is not sustainable. In spite of you are feeding the billion of people, it is not sustainable. You have bring certain good variety, but if this variety has a detrimental impact on the environment, again, it is not sustainable. You have bring some new technology that there is no impact on the environment. You can feed the billion of people, but if there is no profit to the poor farmer, again, it is not sustainable. So as a scientist, we need to think in that perspective, in the idea, what exactly is sustainable? So you need to bring something that there should be no detrimental impact on the planet, or means there is no change on the climate because of this technology. Yes, you can feed the million of people, so you can arrange the bread for these people, you can go for the zero hunger, and moreover, there should be also the profits. If you can tick all these three boxes, definitely that will be the real sustainability in terms of agriculture. Why we need to do that? Why we need to bring that? Because we know that this, from the FAO, that we need to increase this, the global food production almost like 40% by 2030, 70% by 2050. The reason behind that, there is a population burst. And you can see that there is an increase, the population is increasing exponentially. So we need to find some sort of solution for that. And recently, the chairman of UN said that at the beginning of 2020, 135 million people were facing food insecurity. Now we are at 21, and this data was at the end of 2020, that if we are not going to solve this, this is going to increase 265 million. So you imagine how big this problem is there. So we need to find the solution, but again, it should fall on SDG and it has to be sustainable. So who to pick up? Answer is simple: I love this guy, as I told you before. Whether you can say he's my girlfriend, boyfriend, whatever, I mean, I really adore him, because the reason behind that, in spite of suffering, still there is a smile. This guy is providing everything: food, shelter, economy, money, everything, but still he is suffering. Whether it is the biotic stress, the pathogen, or whether it is the abiotic stress, that is the drought or the crop loss. So still, it's our responsibility, being the, this young generation, it's our responsibility to protect these guys. And there are the number of ways you can protect these crops. And again, I'm telling you, I'm not saying that this technology is good or bad. The only thing I'm trying to preach that the technology which we are going to bring is totally sustainable. We know that you can breed for resistance. We know that, I mean, this phenomenon for a pretty longer time. I mean, we can, breed for resistance gene, but you know that it does take like more than 6 to 8 years. And I think the Dr. Sharma know very well, because you need to bring the homozygous, because you just want one resistant gene into the elite variety, and rest of the genome you want to keep the same of the elite variety. So you need to cross several times so that your homozygosity should be maintained in the elite crop, and there should be introgression of one gene. So it's a kind of long time taking process. Next is transgenic. We are in the era of these GMOs and all this stuff, but trust me, now there is a big problem. I can't talk about, maybe some other time I will give a talk on GMO, but at least today you should understand that in transgenic, it is not only science, there is a ethics, society, and the policy, or the politics, I would say. So it is not very easy to take this forward. And I can tell you very honestly, once I was giving a talk back home in London, and the people were very offensive if you talk about transgenic or the GMOs. We modified to jack, that is called as genetically engineered crop, by using the new technology called the CRISPR-Cas. But again, there is a problem because of transfer of the vector into these crop, and again, there is a debate is going on whether it could be considered as a transgenic or not. Third thing is that, it's a long practice. We are using the chemical control, like the pesticides or the insecticides for these crops, and we got protection. There is no problem in that. But what is the result after 3, 4, or 5 years? The soil is polluted, there is a leaching, you can disturb the biodiversity, and finally it is detrimental to the environment, and because of that there is a lot of new calamities every day coming to the environment. So, क्या करें? कैसे करें? ढेर सारी राहें हैं, 3 चीजें. So, as I told you before, there is a number of ways you can do this. But what I would like to say that always new technology will be fruitful if it is user friendly. If it is not user friendly, even if it is mobile, if you can develop any app, if it is not user friendly, it is not going to work, it is not going to sustain. It has to be user friendly. And what we have done, we have exploited the plant natural defense. I would say, very seriously, you don't need to do anything to the plant. They are smart guys. Kuch nahi karna aapne. You don't need to do anything. What we have done, we simply make the plant aware, hey guys, you have a well-defined defense system. Aur humne kuch nahi kiya. And I will explain you how does it work. Simply, हमने ये बताया पौधों को कि you guys have a very well-defined system. You don't need to do anything. And if you do this, what we have done? We didn't use any insecticide. We didn't use any pesticide. There is no GMOs. There is no JAG. There is nothing extraordinary fancy work. We simply try to make the plant that you have a well-defined system and you should take into consideration. And then the question was there, how to do that? Do this plant have the memory? And again, don't confuse with the cross tolerance. I'm not talking here about the cross tolerance. We are talking about, do the plant have the memory? As in the case of the animals or the humans, we have a very well-defined immune system, like the T cells, B cells, memory cells. It means once you are attacked by any pathogen, it could be guided to the different cells, and it could be stored as a data bank or the small USB that, yes, you are attacked by this pathogen. And if you are going to attack by the pathogen again, then there is a battery of arsenals will come forward and they will combat the disease. But in the case of plant, unfortunately, they don't have any well-defined immune system. When I say well-defined immune system, their immune system is very well developed, but they don't have the well-defined immune cells. They don't have T cell, B cell, helper cells, memory cells. In fact, every plant cell could act as an immune cell. So imagine how beautiful is that during the evolution. I mean, how beautifully they have evolved, to combat this pathogen. And this is the reason most of the plant are resistant with most of the pathogen. Otherwise, we are not. If there is any change in the atmosphere, we are attacked, we have a pathogen, again we have a disease, after 3 days again we have a disease. But in the case of the plant, they are resistant with most of the pathogen. And what we call this memory, we call this memory as priming, and the whole talk will be based on that. So just try to focus on this slide, and it will give you the exact message what I wanted to deliver today. So imagine this is your normal plant, and when this plant is going to attack by any pathogen, any biotic stress, there is a induced resistance. We know that, and once there is a resistance, there is a defense response. But what this happens? So this is a well-known phenomenon, and that's what I said in sometime in science. It is not important to bring a lot of data. It is not important to bring a rocket science. Idea has to be novel. And if the idea has to be novel, it is very easy to sell. When I say it is easy to sell, means you can easily publish into the high impact journals, into the, and share your data at the international community. So if this plant is attacked here, so there is a induced resistance. But what happens once this is attacked? There is a signal that will be translocated to the distal part, and the plant sense, oh, I was attacked. And once they sense this, they bring into the alarmed state, and this alarmed state or the vigilant state of the plant, what we call as the primed state. So the plant can sense during the induced resistance, and once they sense this, they bring into the alarmed state, and what we call this we call as the priming. And you can see that I use the same photograph of the leaf just to make sure that there is no allocation of photosynthate during the the priming. Because the problem is that if you bring a super resistant variety, yes, it is resistant, but most of the photosynthate will go to the defense response, and there is a yield penalty. And as an agricultural scientist, we don't want to bring any crop or create any crop that this is super resistant, but the yield is low. Here, what is happening? Exactly, there is no big change in terms of allocation. If the plant is in the primed state, or it is in the alarmed state, or it is in the vigilant state. I can tell you a very simple analogy. Suppose you are in your college, and if you know that dean will visit you, you will be very active. You are not doing anything, but in your office, you can make your office clean very nicely, books are arranged nicely, something like that. Imagine there is one more faculty. He or she doesn't know that the dean will visit today. He is very relaxed. The other guy who was vigilant, he's prepared, but he's not doing anything. He is simply prepared, he is simply vigilant, he is simply active. It is not spending any of his energy still. And when the vice chancellor will visit or the dean will visit, he is prepared and he can give a very nice talk. Other guy can't, in spite of he was good. So similarly, in the case of prime, once the plants are primed, they are in the vigilant state. And if they are going to face the next stimulus or the triggering stimulus, this priming boosts the plant defensive capacity. And as I told you before, definitely it can bring the plant into the alarmed state. And this is the beauty of the priming. No resistant variety, no insecticide, no pesticide. Simply, you are using the plant to bring into the alarmed state. And moreover, I use the word the vaccination. What happened in the vaccination? You use the same antigen and the body developed the antibody. So what we do in the priming, we use the same pathogen that cause a disease, but this is regulated. So you need to make sure that this pathogen could not behave as a stress. So this is the thin line that you need to maintain and make sure that this could not be stress. You can use a mild dose of this pathogen just to make the plant aware, hey, come on, you are attacked. And once they are attacked, you are in the primed state, and if the plant is going to face the same challenge, then there is a boosted defense response. So that is the beauty of priming. I can show you if I have a picture. Yes, so definitely that suffices the story that the plant do remember stress. This is one of the model plant Arabidopsis. I think some of you know, some of you might know, but yeah, this is a very the model plant, because the life cycle of plant is 1 year, 1 month. Seed to seed is just 1 month. So you can study any physiology, transcriptomics, proteomics, metabolomics in this small guy, and it is dicot. So it's very linked with this, the higher plants. So what happens here? If this plant is attacked by any pathogen, let's say here is spider mites, there's two things. This is the plant defense hormone, and this is the downstream signaling. It means there is a activated defense response. So there are two components. One is the defense hormone, and one is the downstream signal. So they will activate, we know that is the induced resistance. During this, now the signal will be translocated to the distal parts. So in this part, what happens? They are in the primed state. It means they are ready with the arsenals, but they didn't fire the bullets. It means still there is a no cost. And imagine how beautiful is that. So if there is no pathogen, why plant need to activate its defense response? But they are ready. Means the hormone, the defense hormones is ready, all the bullets are ready, all the guns are ready, but still there is no firing. However, if they are going to attack this guy, then there is a activated defense response or the boosted defense response, and that is the beauty of the priming, and that is the real sustainable approach for the crop protection. And yes, so you can clearly see that. So this is your defense intensity. You can bring the priming stimulus here, and then after that, if it is attacked by the pathogen, you can see that how strong defense intensity have been increased because of the prior experience of any mild stimuli. Moreover, I can't talk so much. There are a number of ways you can bring the plant into the primed state. As I told you before, the pathogen, even the root microbes, that is, they lead to the ISR, the PGPR, the plant growth, the promoting rhizobacteria, the volatiles, and even certain synthetic chemicals, as well as the biological elicitors. So there are the different ways you can bring the plant into this the defense priming or in the alarmed state. And that's why I said is that it is an adaptive part of induced resistance. It means the plant has adopted something. Plant has gained something. Because of this gain, they can sensitize something during the induced resistance. Because of this sensitization, they are in the alarmed state, and that's the reason they are more vigilant. And if they are going to face the challenge again, they are super protected. And indeed, I say that it is intentional. We are creating the priming in the plant. It is intentional but regulated. If it is not regulated, the same stimulus could act as a stress and there will be no priming. So you need to really understand the thin line between the stress and the stimulus. The same stimulus could be stress if it is not regulated. So that's why it is called as intentional but the regulated use. Now the second question is there, what could be the best stage to prime the plants? In my knowledge, we have done a lot of experiments. Even we have also published a number of work that the priming could be done at the seed stage, the flowering stage, rooted stage, branching stage, and in more or less, almost in everywhere, we can induce the priming and we can show the production. I can just show you an example. One is that seed stage when I was in the Lancaster, Britain. So simply take the tomato seeds. These tomato seeds were treated with the jasmonic acid as an elicitors. After this, the seeds were planted, tomato plants grown, and then they were challenged with Botrytis cinerea. This is one of the pathogens that cause a disease. And these plants were super protected. So so definitely you can prime the plant even at the seed stage. Now, the big question was that how long this priming could last. So suppose if I prime the plant today, will the plant will be primed after 1 day, 2 days, 3 days, or after 3 days the plant again back to normal? So this was the biggest question that we wanted to answer, that how durable is this defense priming? If the priming is done, it is long lasting, it is short lived, it is stable, it is unstable. So that was the one question that we wanted to answer. And I will be very happy to share that we have published number of articles. So you can see again here, that is the priming, how long this priming could be maintained, or the maintenance before the attack. And I say this is the week after treatment. So you can see there are I have more examples, but because of time shortage, I can't talk more. But yes, even after 5 weeks, 6 weeks, or even after 2 months, if the plant is primed, they are still vigilant. And if they are challenged with the pathogen again, still their response is more stronger as compared to the non-primed or unprimed plants. So that is the beauty of this the defense priming. So that suggests, yes, indeed, that the priming is durable. It is not short-lived. Yes, it is a long-lived and it's a stable phenomenon. And we know that we call that is like a within generation priming. It means the same plant could be the prime, and as I told you before, so it could be the part of SAR. The priming could be done with biotic, abiotic, herbivore. We can use some sort of the chemical the component. The one of the component is a very pronounced, well-defined the priming agent, that is the beta aminobutyric acid. A lot of work has been published on this, the BABA. Now the second question was that the first question was whether we could establish the priming in the plant or not. So this answer. The second question was that how durable is the priming? We answered that yes, the priming can at the last in the generation. So this is a stable, long-lasting phenomena. The third question was there, could we use abiotic stress to prime the plant and challenge with the biotic stress and see whether there is a protection or not? So that was the third, the big question that we wanted to answer. And this what we published long ago, in like 6 years ago in the Plant Cell, one of the reputed journal. What we have done here, again, I told you, again, sometimes the concept has to be novel. We didn't do any extraordinary. Again, the concept was the same, whether the plant do have the memory. So what we have done, so this is Arabidopsis. So this plant, what we have done, we have kept the plant at, normally Arabidopsis grow at 24 to 25 degrees centigrade. We kept the plant at 28 degrees centigrade for 15 to 20 minutes. Just to let them feel that they are facing some stimulus, but not stress. And again, we bring back the plant to the normal growth condition. And what we have done a repeated, just to make sure that the plant can sense, oh yeah, they could face this, the challenge or the stimulus again and again. And if they feel that, they should be in the primed state. All right, now we have to be in the vigilant state. And what we have done 1 day, 2 day, 3 day, 4 day, like for 1 week. And I'm showing you the data just because of the heat stimulus. But in this paper, we have done for both heat stimulus, salt stimulus, as well as the cold stimulus. And again, I'm using the same photograph just to make you understand in the primed state, still there is no big allocation of photosynthate and the plant is still growing normally. There is no yield penalty. And after that, it was challenged with this, the biotic stress. And the beauty is that, imagine, we were so happy and surprised that this is H0 means no stimulus, 1 day, 5 day, and even after 7 days, we were able to establish the priming. And after 7 days, if this plant was challenged with one of the bacteria that is called as Pseudomonas syringae, one of this the pathogen that caused the disease in Arabidopsis, and these plants were super protected. So again, that suffices the story that yes, indeed, the priming is long-lived, and the priming could be biotic, abiotic, biotic. Moreover, again, if you prime the plant with one stimulus, it is a broad spectrum resistance. Again, I will try to explain in a very simple analogy. If you are vigilant, doesn't matter, vice chancellor is visiting you, head of department is visiting you, dean is visiting you, student is visiting you. You are very vigilant. So this is the second important beauty of that. If I prime the plant with Pseudomonas, plants are protected against Pseudomonas, but this is also protected against botrytis. This is also protected against some more stresses. So that's why it is a broad spectrum resistance in terms of agriculture productivity or in terms of agriculture sustainability. So I think I have already explained you that if the plants are primed, the response is faster. Response is earlier and more sensitive. More sensitive means, I can tell you a very simple example. In the agricultural field, the plants are growing very normally. There is a very nice sunlight. And again, there is a rain. Because of high humidity, just after 2 days, whole crop is vanished. There is a bacterial infection. But imagine if the plant is primed. Even if there is a bit of stimulus in the environment, they can sense. But the normal plant can sense, they can only sense once they are attacked. That is called induced resistance. And if the induced resistance is stronger, the plant can still protected. But if the induced resistance is not stronger or the pathogen can breach the IR, definitely there could be a disease. But in the case of priming, even if there is a mild dose of stimulus in the environment, the plant can sense that and they can bring into the vigilant state before the triggering stimulus comes again. And that's why the response is more sensitive. They can sense more earlier, quicker, faster, and that's why the strong is more response. So that is the fourth the important observation from the priming. Now, this is one of the most. Again, I'm talking about maintenance. If I'm vaccinated, my son need to be vaccinated. Simple, जैसे अभी कोरोना चल रहा है, अगर मेरे को कोरोना का वैक्सीनेशन लगा तो घर में सबको लगेगा. Similarly, if I have a vaccination of the polio, if my dad has a vaccinated polio, I also got vaccinated and my son will also be vaccinated. So it means this vaccination will not go into the next generation. And I use the term vaccination Green vaccination means protection of the green plant or the plant. So now it was $1 billion question. Could this priming can go into the next generation? Till now, we have just discussed that it will go into just one generation within generation. Now the question was there, could this priming go into the next generation? Could the plant is able to maintain this priming in the seed? And again, I can't talk about the mechanism because there is a specific mechanism behind the priming, and mostly it is epigenetics. It is not genetics. So there is a lot of changes in the histone methylome or the the methylome or the histone acetylation, demethylation, and these are the reason that could lead to go to the next generation. So that was the biggest question for us. We call that between generation priming. Mean दो जनरेशन के बीच में प्राइमिंग संभव है कि नहीं. अभी तो एक जो पौधा था वो प्राइम हो गया. अगर वो पौधा प्राइम हो गया, उसकी हमने सीड कलेक्ट कर ली, फिर उसको सीड को हमने फील्ड में ग्रो कर दिया, अब वो प्राइम होता है या नहीं होता है, या हमें फिर से स्टिमुलस देना होगा. Like, can parental stress increases offspring resistance, and we call as transgenerational immune priming. So that was the next question that we wanted to answer. Could we establish this? If we could establish this, definitely there is a big impact on the agribusiness, and we can talk about the more on this, the companies with the seed or something like that. So we call as TGIP. So I mean, we have done with a number of pathogens, but I will just show you one or two examples just to make this story clear. Look here. So this is your plant. This is your control plant. So imagine we are dealing with 3 generation 1, 2, and 3. We are just dealing with 3 generation. So this is grandparent, this is parent, and this is offspring. So now we are dealing with 3 generation, grandparent, parent, and the offspring. Grandparent was challenged with the PST, and they were primed. And we collected the seed from the grandparent, and what we have done, we divided the seed into 2 parts. In one part, we just grow the seed as such to have the next generation. In the second case, again, we challenge the seed with the PST and grow the seed. And again, you have the plant, and again, you collect the seed, and you can grow the seed in this 3rd generation, and again, you can grow the seed in the 3rd generation. So in this case, if you look here, only grandparent was primed or challenged with the stimulus. Parent was not challenged with the stimulus, and definitely offspring was not challenged. In other case, both the grandparent and the parent was challenged with the stimulus, the offspring was not challenged. You know what important observation, the beauty we found that in both cases, I mean, this is one of the examples of this, the downy mildew. In both the cases, they were protected. It means if the grandparent is challenged, parent is resistant, and offspring is resistant, it means this priming can go to the next generation. And yes, indeed, that is the beauty of that. And I can tell you very confidently, in my hand, I have tested 8 generation in Arabidopsis, 4 generation in barley, and now we are testing more in a BHU, and we are getting the same sort of significant result in terms of the protection. So definitely, yes, indeed, this priming can last for next generation. Yes, we have already published this work. I'm not showing all my research work, but some of the things that could be more relevant to the slides. So we have published this, and again, it was related to the Jasmonic acid. And yes, we have also tested with some more the biotic stress. So instead of the pseudomonas, we have used some spider mites. We have also used some other stress. And again, in both the cases, the transgenerational priming was easily established. And you can clearly see here the number of eggs laid by the spider mites were less because of prime plant. And the reason behind that, these plants have more jasmonate, and these spider mite doesn't like to lay their egg if the leaf has more jasmonate. So this is again linked with this the priming phenomenon. And yes, I just try to give an overview, but there is a lot of mechanism going behind that. So there is a clear-cut omics data on that. So we have done transcriptomics, we have done metabolomics, we have also done the proteomics. I can explain you more on that, but I can simply show you here. There's a lot of changes takes place during priming phase, the post-challenge prime phase, or the transgenerational the prime state. So like the changes in the metabolites, in the amino acids, the glucosinolates, the PR that is called as the pathogenesis related gene, the change in the defense hormone, jasmonic, SAR, as I told you before, the histone modification. I mean, there is a lot of changes. So you need to also monitor what are the changes taking place during the priming, what has been in the post-challenge, and what exactly lying in this the transgenerational prime stage. So these are the five points that we discussed: the priming, establishment of priming, long-lasting priming, transgenerational priming, and their mechanism. Moreover, we also try to link the priming with one of the more, again, important aspect in the case of agriculture, is called as seed dormancy. We know that this is one of the important adaptive trait that could have a major influence on the plant performance. And what we found, if the plants are primed, they are less dormant. And again, it is the beauty. So it means the biotic stress also impact the seed dormancy. So if you think in terms of ecological implication, if the situation is going to be stressful in the environment, the plant try to germinate quickly and establish, and they can bypass this stress. And once they can establish, they can feel the stress. So that's the reason, if the plants are primed, the seed can grow quickly as compared to the other one. And once the seed can grow, they can establish themselves. And once established, even there is a, like the stress, they can easily withstand. But imagine the other case, if the seed is not primed, or it has a, like the dormant period, they could, it's a very natural phenomenon. They could bypass the seed dormancy, but again, they have to face this challenge again and again. They can establish. But once the plants are primed, they can easily face this challenge, and again, they can go on that. So now we are also working some more pathogen with the seed dormancy. And right now, one of my students, she is working with of the very well established the biostimulants, or the biothing that is called as the Trichoderma. So we know that the Trichoderma could be very useful in number of aspects. So we are trying to establish whether we could use Trichoderma as a priming agent or not. So these are the different stages of the Trichoderma that you can see that. And the preliminary result shows that yes, indeed, if the plants are challenged with the Trichoderma again, the plants have the less dormant, and the seed can grow quickly as compared to the norm. So now, look, we started the story with the priming, we ended with the lasting, we ended with the TGIP, and we ended now with the seed dormancy. And now that is real sustainability in terms of agriculture, what I was discussing you before. And that's why I like these slides. So stress parents, result in the stronger offspring. So if you are a little bit stressed, your efficiency is always increases. If you have to submit any proposal, you are a bit stressed, your efficiency is increased. If you are a student, before a day examination, you are a bit of stress, your efficiency will be increases. Similarly, in the case of the plant, if you are a bit stressed, stress means, again, I'm using the term stimulus. Because of this stimulus, if you are in the vigilant state, imagine karo as a student, जब एग्जाम होता है, you are very vigilant. किससे नोट लेनी है, किस टीचर से बात करनी है, क्या करनी है, कैसे करना है, and then your productivity will be easily evaluated, and you are more productive. And that's why I said this stress parent results in the stronger offspring. Now see the profit. So we have discussed two aspects: people and the planet. So we saw that it could be plants could be easily protected. There is no detrimental impact to the environment. Now see the profit, and I have already explained indirectly, in the case of the farmers, if they have own seed stock of these varieties, they can keep for the longer period, and they were less vulnerable to any outbreak of the pest and disease. We already established that indeed, yes, it's durable, and once it is durable, it can maintain throughout the life, and it could be going to the next generation. And again, as I told you before, the mechanism is epigenetics. Moreover, I'm not saying that you should not use insecticide and pesticides for the full protection, but what you can use, what you can do, you can minimize. Imagine if I'm using 1 liter of pesticide for the crop protection, just use 1 milliliter. So if you have the prime crop, and if you use just 1 milliliter of pesticide and you are getting the same protection, bingo. So again, I'm not saying that you should not use insecticide or pesticide, but what you can do, you can slowly minimize the pesticide. And if you do this, then there will be a less risk to the environment. And the environment, and it will be more, excuse me, and it will be more eco-friendly with the same protection. So if I try to summarize everything, what I wanted to explain, so we have a different control strategy for the agricultural crops, like monitoring, the pesticides, the rotation, the hygiene, there's the biocontrol, the resistant control. If you let this guy sits here, the priming concept of the priming or the TGIP, and if you use in the right combination, that could be the very good strategies for the non-pesticide IPM. And that could be easily sustainable. And that's why I could confidently say that indeed this priming is more sustainable, because it is ticking all the 3 boxes. People, you have crops are protected, planet, no detrimental impact to the environment, as well as you also have the profit. And that's why it is a broad spectrum effectiveness. And the take-home message is that, yes, it does indeed boost the plant immunity, green vaccination, and yes, it is the sustainable approach to the crop protection. Moreover, if I will say that, we have a different station. When I was in Britain, we have some of the station back in, the Cape Town, the Africa, and now you can see that these farmers were super happy with their own crops, and they can, easily sustain with the priming phenomena. So what is that? You can grow, sow, reap, and repeat. So अपना पौधा ग्रो करिए, सीड को सो करिए, उसको काटिए, फिर से ग्रो करिए. And still you have your own seed stock that are less vulnerable to pest and the pathogen. And that's why that is the real the sustainable technology, I would say, in terms of sustainable agriculture. And I think this is the last slide. I would say that, yes, so we know that he is Albert Einstein. He was smart. We know that there's a number of ways you can protect the plant healthcare. So if you use the plant defense priming, it is smart plant healthcare. It means, yes, you are protecting the plant, but this is the smart way to protect the plant, because you are clicking all the 3 P's that is required for sustainable agriculture. And that's why आज की date में अभी जो, even there is a big abhiyan going on in our the back home in India in 21, even the Modi said that Atmanirbhartaye Vishwa Kalyanam Cha Vigyanam. So that nicely again fits this concept of defense priming, that is science for self-reliant. It means you are self-reliant and for global welfare. So again, it fits nicely with the message that we are, they're taking in 2021 in terms of science, agriculture, and technology. So I think I will stop here. I can talk more, but now because of limited time, I will stop here. But before I stop, I would like to acknowledge, some of the guys. I mean, there is a lot of list, but I mean, these are some of the guys whom I work, and still they are my collaborators, like the Mike in Lancaster, the Madan in the States, Jimmer in Taiwan, Christine back in Texas. And these are the funding agencies like NSC, USC and IFA, BBSRC. Since I came back to India, we got the funding from UGC, and now I also got the funding of 35 lakhs from, because the BHU has got a reputation of Institute of Eminence, BHU IOU, so I also got the grant from IOU. So still I'm working on this, taking the priming and try to interact with local farmers more. And that is the real satisfaction of your research work, when you see the research work in the reality, because sometimes what you are working in the lab doesn't work. So you need to bring the idea lab to the field, and that is the real work. Yes, I mean, if somebody would like to contact me, I mean, these are some of my details, whether it is email, Twitter, mobile, or this like the profile. You can easily reach me in there. So now, because of this, now I can stop this, and I hope the message is clear and it would rejuvenate more. And yes, indeed, if I get more chance, in the future, we will talk some more ideas and the more research and for the Parul University, and bring more sustainability to the agriculture. So again, thank you very much, Dr. Sharma and Dr. Vikram, and all the participants, those who are joined either with the Google Meet or through the Facebook or the Instagram to hear me. I hope the message is clear, but still, if it is not clear, I will be very happy to answer in person, offline, online. Thank you so much again.
Okay, sir. Thank you, sir. Thank you so much, sir, for this valuable speech, and give a nice information about plant defense priming. Thank you very much, sir.
Thank you.
Dr. Prashant, I have a small query.
Yes, please go ahead.
No doubt, you have explained the things fabulously, and I think I am very much sure that my students, those who are listening on the different platforms, as well as the participants, those who are on the different platforms, they have got the benefit. The query is, no doubt, this priming process is related to the upregulation of the genes or defense-regulated genes. We can say that. This priming is more helpful in the case of the
Priming will not lead to upregulation of gene, because once it is lead to upregulation of gene, then there is a defense response. Priming will simply bring the plant into the alarm state.
Very true, very true. But there are some literature which quotes regarding the priming, how it is related with the upregulation of the defense-related genes. The question is whether it is more feasible in the case of the abiotic stress or the biotic stress.
Well, very good point, right? So to be very frank, I mean, the most of the data which I have shown you is the biotic stress. What we have done recently, even in the PSU, so we have used the abiotic stress. So what we have done, again, it's a very simple experiment. There were the two pots, right? In both the pots, they were watered nicely, okay? And after that, one pot, we kept them for drought. Like for 3-4 days, there is no watering, and the other plant was, giving them water, so them. So what we found in the plant where we kept them the drought, they were in the primed state. And what we have done during the primed state, we have checked their stomatal conductance, and we've also checked the stomata. So most of the stomats were closed, and moreover, we have measured the ABA content. The ABA content was higher in the case of this drought prime plant. And what we have done again after that, so the non-drought plant, they were completely gone, okay? And if you water them again, still they won't revive. And for this, the prime drought, you don't need to water them, still they can grow very well. So I mean, this is the first experiment, and to be very frank, I'm very happy to take this on the drought, because that is more feasible in terms of day-to-day scenario, in this like this belt, the kind of this belt. So, but yes, the preliminary data strongly suggests that yes, indeed, the plant could be also primed with the abiotic stress, specifically as I told you, just with that.
Dr. Prashant, as you have quoted, yes, it is working in the case of the abiotic stress. But if we see how it is transferring from one generation to another generation, it is easy to carry some information which is related to the biotic stress. So how it is beneficial in the case of the biotic or the abiotic?
I mean, in terms of the biotic stress, as I told you before, it is not the generics. So imagine if we have some drought resistance prime plant, and if you want, imagine if there is some barren land where we need so much of water. If we use the same crop, let's say the rice crop, where the requirement of water is very, very more, right? But still, if we have these guys, I'm not saying that they don't require water, but we can minimize the amount of water. Instead of 1 liter, they can just take 10 liter of water, and still we have the same kind of the seed set or the protein. So in that scenario, that could be, the beneficial. And in terms of the next generation, in terms of the biotic stress, I can tell you very confidently, because there are the certain epiloci markers during the seed set. These markers will move from the vegetative stage to the reproductive stage, and we have worked a bit of this, the small RNA, and they can also take part in this, the transferring of this epigenetic during the seed set. Similarly, I assume that in the case of the drought, there also be some mechanism, maybe the epigenetic, that will be passed from the vegetative stage during the seed stage when the plants are primed, and that could be responsible why it will go to the next generation. But yes, that need to be, dissected more, because right now I don't have any the scientific data to prove that.
Thank you very much. Thank you very much.
Thank you so much again. Thank you very much. Again, it's a great honor to be the part of the Parul University. And again, the Professor Sharma, I will be very happy professionally, personally, directly, indirectly, if I associated with this, the private renowned university in terms of ranking, right? So I will be very happy if there is any collaboration or any kind of this thing from my side. It's totally open.
Sure,
Sir, wherever
sure.
you are free, hello, Sir Arpan Naik, wherever you are free, then visit. If you are visiting Baroda, then please come. We are welcome.
Sure, absolutely. Because of the COVID now, the things have been so restricted. But definitely, any time the Sharma will call, I will never say no to him. So there is no chance that I would say no to him. So definitely, I would like to visit. Absolutely, yes. And there will be more interaction, person to person with the student and even with the faculties.
Yes, sir. Thank you so much for your valuable time, sir. Thank you. Thanks a lot. Yeah.
Dr. Vikram.
Namaskar again. Thank you so much. All the best. Thank you.
Thank you.